Good morning.
Good morning. How are you two doing?
I'm doing fine. Are you?
Good, good. Thanks. Looks like it's just the three of us for now. So let's get started. So there is the review package that I post online. If you haven't got a chance to get it, you can get it from eCentennial. So I'll just be going through these questions. But before we get started, since it's only the three of us, are there specific topics you would like me to go through? Navleen or Yusuf, any specific topics?
Not really.
Not really? Okay, so I'm just going to go through these questions and feel free to ask if you want me to elaborate. Okay, so the test is going to be lecture 2, 3, 4, 5, and plus the lab. So I will start by reviewing lecture 2 right here. Okay, so macromolecules. First question, what holds the atoms together in a water molecule? How would you describe a water molecule? What interaction exists between two adjacent water molecules? So it's a lot to unpack here. Water molecule, as you all know, is H2O. Okay, so I'm gonna write a chemical formula H2O. So what is a molecule, right? A molecule is basically atoms coming together in a specific ratio, and then you're gonna form a molecule. So there are two types of molecules that we learned. There's the ionic molecule, and then there is the molecular compound, right? So H2O is the covalent compound or molecular compound. And if you draw it out, I'm sure you learned this in chemistry already. This is what a water molecule looks like. So what holds the atoms together in a water molecule? Well, this here, that is a covalent bond. Covalent bond. And a covalent bond involves the sharing of electrons. Okay. Now, how would you describe a water molecule? Overall, the water molecule is neutral. Okay, neutral. Neutral means no charge, right? In chemistry, I believe you probably have learned about things like, sulfate, SO4, 2 negative, right? So that is a charged molecule because overall, there is an imbalance between the number of electrons and the number of protons. So you have an overall charge of 2 negative, okay? Meaning you have 2 extra electrons in the entire molecule. But as for the water molecule, it is neutral, okay? But we will say that it is polar. Polar. So polar means uneven distribution. Uneven distribution of electrons. Okay, so the oxygen and the hydrogen, even though they're sharing electrons, but the oxygen, as we learned in lecture 5, has higher electronegativity, right, compared to hydrogen. So it's going to pull the electron towards itself, making it slightly negative. That's what this symbol is. So slightly positive here for the hydrogen, slightly negative for the oxygen. Okay, so if the question asks you, what's the most appropriate way of describing water molecule, right? The correct answer is that they are neutral and they are polar. So what happens if you put two water molecules next to each other, right? So if I have a water molecule here, and then I put another one next to it, right? Like that, what's gonna happen? Well, the slightly positive hydrogen of one water molecule will be attracted to the slightly negative oxygen on the other molecule, right? And this is going to create some kind of force of attraction between them. Okay, so this force is called, anybody know what that is called? This interaction between two water molecules. No? Okay. That's okay. This is the hydrogen bond. Hydrogen bond. Okay. So hydrogen bond is an interaction between two molecules. Okay, we'll see hydrogen bond again later on, but that's basically what water is. Okay. Moving on. Next question. If you supply radioactive nitrogen atoms, and you know, in case you're not sure what radioactive means, it's something that you can trace and detect. And you know, you supply these radioactive nitrogen to a cell and then you let it grow. What molecules would you expect to find the radioactive label in? So this question basically asks you, where do you find nitrogen in the in macromolecules? So you remember we have 4 macromolecules, right? Carbohydrates. We have proteins. We have lipids.
Lipid.
Yeah, thank you. And what's the last one?
I think nucleic acid also.
Yeah, nucleic acid. Thank you. Okay, so let's write down what they're made up of, right? So for carbohydrates, we have C, H, and O. For proteins, same thing, but we also have nitrogen, right? For lipids, C, H, and O. And for nucleic acid, we have phosphorus as well. Okay, so I should remind you that proteins, there are trace of sulfur as well. Okay, so if you radioactively label the nitrogen, it is going to show up in the proteins as well as the nucleic acid. Okay, any question? All right, so if I radioactively label the phosphorus, then it will show up in the DNA, right? It will show up in the RNA, it will show up in the DNA as well. Okay, so that's that. Next question. Regarding DNA, what is the monomer of DNA? DNA monomer. Okay, so that is going to be a nucleotide. Nucleotide. Okay. What is a nucleotide made up of? Well, a nucleotide has 3 parts, right? There is the phosphate, the PO4 -3. There is going to be a sugar. Right? I can put equal here, plus a sugar. If we are talking about DNA, then it is deoxyribose. Okay? If we're talking about RNA, then it's going to be like an oxyribose, right? And finally, we have a base, a nitrogen base, nitrogenous base, sometimes it's called nitrogen base. Okay? So for DNA, the nitrogen base could be A, T, C, or G. Okay, so these 3 things together makes up a nucleotide. Okay, so you have the phosphate here, right? You're gonna have your sugar, and then you're gonna have some kind of nitrogen base. Okay, so the nitrogen base could be like 1 ring, right? Or it could be 2 rings, right? Depending on what type it is. Okay, so that's a nucleotide. There are 4 types of nucleotides for DNA. Okay. Next one. How are nitrogenous base or nitrogen base classified? Okay, so nitrogen base or nitrogenous base, we can have purines. Okay, purines are A and G. Okay, adenine and guanine. Okay, so these ones, the adenine and the guanine, they have 2 rings in their structure. Okay, so like something like this, right? Like you don't have to draw it, but I'm just showing you what it would look like if you were to draw it out. Okay, so like you would have something like this, double ring. Okay, like an annex here, and that would be a guanine, adenine rather. Okay, so 2 rings, and then the other type is the pyrimidine. Pyrimidine. Okay, so the pyrimidines are going to be thymine plus or cytosine. Okay, so unlike the purines, the pyrimidine only has 1 ring. Okay, so something like this. One ring. Okay. So 2 rings and 1 ring. Now, the number of rings is important for the next questions. But so far, any question? Do you understand what I'm doing here? Okay. Part C. What is the base pairing rule? Okay, so base pairing rule, if you have a strand of DNA, as you know, it's double-stranded. So if one side is A, T, C, G, what's it gonna be on the other side? Well, A always pairs with T, T pairs with A, C pairs with G, and G pairs with C. So in general, purine always pair with a pyrimidine. Okay. And sorry, pyrimidine. I think I spelled it wrong. Okay. And so A and T goes together, and C and G goes together. Okay. What force holds the nitrogen base together along the length of the DNA? Okay, so here is the ATCG, and here's the TAGC. Right, so what holds them together, right? Well, it's the same interaction that we saw with the water molecule. Okay, so between a thymine and an adenine, there will be 2 hydrogen bonds. Okay, between the C and the G, there will be 3 hydrogen bonds. Okay, so 2 hydrogen bonds hold A and T together, whereas there will be 3 hydrogen bonds. 3 hydrogen bonds, which will hold G and C together. Okay, so it is easier to pull apart a DNA that has a lot of A and T's in it compared to something that has a lot of G and C. Okay, easier to pull apart because 2 bonds is weaker than 3 bonds, right? That's going to be important for something we'll learn later. What is the significance of the base pairing rule? Okay, so why is it necessary to have A and T together and G and C together? Well, as I mentioned earlier, the A and the G, they are 2 rings. Okay, so these are 2 rings, right? 2 rings. Whereas the T and the C, they are going to be 1 ring. So across the DNA, it will always be like 3 rings across. Okay, so the base pairing rule ensures the width, right? How far across along the DNA remains the same. Okay, think of like a ladder, right? A ladder. You want the rungs to be the same length, right? Okay. You don't really want a ladder that looks like if you have a ladder that's like this, you know, some rungs are thicker, right? Some rungs are shorter. Like that doesn't make sense, right? It's not really good ladder, right? I guess you see those at parks, but like practically speaking, you would have two side bars, and then everything in between would be the same width, right? And that's what happens with the DNA. If I pair up a purine and a pyrimidine together, then I will make sure that it is always 3 rings across. Okay, you don't want to all of a sudden have a purine with another purine, because then that would be 4 rings across, right? And that's too wide. It actually, if something like that happens, as we'll learn later on, that's a signal that tells the cell there is something wrong with the DNA. Maybe there's a mistake, maybe there's a damage, and the cell will initiate some kind of repairing protocol. Okay, so that is that. By the way, all these questions are taken, like I scanned through all the multiple choice questions, and then like for the test bank, and then I come up with these questions based on those multiple choice. So it really is important that you go through these things afterwards. Any question about the DNA stuff before we move on? Okay, number 4. What is dehydration synthesis? Illustrate with a diagram the significance of this process in building a polymer. Okay, so you probably remember polymers and monomers. Right? So these are monomers, little building blocks, monomers. Okay, and we can put them together to form long chains of polymers, right? Polymers. Okay, so from monomers to polymers, we are going to do the dehydration synthesis. Dehydration synthesis. Now you might also see it written as condensation, okay, aka also known as condensation reaction. Okay, so you will generate water molecules, okay, when you link 2 monomers together. And just for completion, if you want to break it down, right, we would have to do the opposite of dehydration synthesis, which is hydrolysis. Okay, so to connect this to, I think lecture 4 or something, right, to build things, this is part of your anabolism. Okay, and in order to do that, you would need to supply energy. So ATP is needed, right? ATP is needed. And then over here, hydrolysis, right? That is part of your catabolism. Okay, and then it's going to release energy, okay, which would then be captured and be stored as ATP. Okay, that is that. Now, illustrate with a diagram. Now, I don't know if you remember, but we did this in the tutorial for number 2, lecture 2. Okay, like I give you a diagram, for example, and let's say I have something like this. Like you don't have to know what that is really, but I'm saying, you know, there's an OH here, and then you know, maybe you're gonna have another molecule here that has an OH, and then I will say, you know, they come together through dehydration synthesis. What would the final product be? So you should know that the OH will be removed from one of them, and then the H will be removed from the other, and then you're going to end up having something like this. Okay, plus H2O. Is that okay? Anybody Have any question about how I get those? Okay. Now slightly harder version of this. You know, you might have like this one, right? We put more space. Okay, let's say you have like a C double bond O, OH, right? And then like a CH3 here. And then over here, you have C, OH, H, right? And you add them together. Again, what you need to do is you need to find the OH on one of them. It doesn't really matter which one you take the OH from. You can take it from here. It's the same thing. But if you take the OH here, you will only take the H here, right? But I took the OH from this thing, and then I'm going to take the H from here. And afterwards, it's going to look like this, right? Afterwards, it's going to look like this. Okay, everything else you just copy, right? So you learn the name of these things in chemistry next semester. Okay. So you can see this part here, right? Sorry, maybe I'll just highlight it. This part here, it's really from this molecule. And then the rest of it is from here, right? And of course, we're going to have plus H2O. Okay, so this is an example of a dehydration synthesis. Okay, you're removing a water molecule. That's why it's called dehydration. Okay. All right, next, regarding enzymes. What is an enzyme? Okay. So an enzyme is, what kind of macromolecules is an enzyme? Anybody know? Is it carbohydrate? Is it protein? Is it lipid? Is it nucleic acid? What is an enzyme? Okay, if you're thinking about protein, that is correct. An enzyme is a protein. Okay, what does it do? It catalyzes reactions, chemical reactions. In the body. Okay, so how does it work? Now, this was discussed in the lecture. This is energy and this is time. So you should know by now, in the chemical reaction, there is some reactant. So let's say A plus B, which will become products, right? So let's say C plus D, and then you will need to have the enzyme, right? Okay, so let's see what happens without the enzyme first. Okay, so you will have A and B here, A plus B, the starting material, and then in order for the reaction to take place, you actually have to supply a little bit of energy, and then it's going to come down like this. So this energy, which you have to supply to kickstart the reaction, that is called the activation energy. Now, if you were here on Friday, you will remember we talked about something like this, something similar to this in glycolysis, right? Glycolysis, which is the splitting of the glucose into 2-pyruvic acid. There is the energy investing phase, and then there's the energy yielding phase, right? So we first must supply 2 ATPs to kind of kickstart the process. So those 2 ATPs would be what's serving as the activation energy. Okay, think of this as opening like an investment account or something. Like you might have to pay some upfront a fee, okay, administrative fee in order to open the account, and then you can use it to, you know, invest and make money. So that's the amount of energy required to kickstart the reaction. Okay, now this is without enzyme. Okay, with the help of enzyme, you will be doing the same thing, but now the activation energy is much lower. Okay, so enzyme lowers the activation energy. Okay, think of another analogy is like tobogganing. Okay, you got to go up the big hill before you slide down and have fun. But with the help of an enzyme, you don't have to climb as steep of a hill. You just climb a small hill. You can still slide down and have fun. Okay, so that's what enzyme does. Okay, it lowers the activation energy. Okay, next one. What factors affect the functionality of an enzyme? Well, since enzyme is a protein, so anything that affects the protein is going to affect functionality. So things like, pH, right? If you change the pH, if you make the environment more acidic, more basic, that's going to affect your enzyme functionality. It could be ionic concentration. Right? If you change the amount of ions, such as NaCl in the solution, that's going to affect your protein. Temperature, definitely. Right? Temperature. Type of solvent. Okay, so all these are things that would affect the functionality of the enzyme. Okay. How does the enzyme lactase work? I guess that's what I try to type here. Work. Okay. So lactase, right? Okay. So here is lactose. Okay. So lactase is the enzyme that will break down lactose. What is lactose made up of? Lactose is made up of glucose plus galactose. Okay, so you remember that the enzyme is going to have like an active site, right? Active site. And that will allow the substrate to fit in it, and then there is the bond that's holding the glucose and the galactose together, and the enzyme will break that bond, and then you will release the glucose and the galactose like that. Okay, so that's what the enzyme does. All right, moving on. Complete the following chart. Okay, we'll see how this goes. I don't know why it's not. Can I make it bigger now? Okay, there we go. Okay, so monomers. What are the monomers of carbohydrates? Anybody? Monomers of carbohydrates? Any examples of monomers that you remember? Yeah, go ahead, Yusuf.
Is that monosaccharide?
That's right. So there are the monosaccharides. Very good. Monosaccharides. Okay, and I'm just going to list some here. So they're glucose, fructose, galactose, right? Okay. Those are the monomers. Biological functions. Energy source. We also use them for structural support. Okay. Primary energy source for many, or for humans, definitely. So examples would be starch, glycogen. Right? So these are storage form of energy. Structural support would be things like cellulose. And we also talk about something called chitin, which forms the exoskeleton of insects, for example. That is that. Proteins, monomers of proteins, amino acid, amino acids. And there are 20 types, right? Okay. I should probably mention essential ones, or 8 essential ones, which means you cannot make yourself. You must consume it. You must eat it from your diet. Biological functions of proteins. Well, there are a lot of functions for proteins. Structural support. Okay, like your hair, your nails, horns, that kind of stuff. All those are proteins. We need them as enzymes to catalyze reactions. Oh, we should say catalyze reaction. Okay, signaling in the body, right? Transport. So examples, we would have enzymes, right? Enzymes. So we talked about an enzyme earlier, lactase, which breaks down lactose. Hormones for signaling. So an example would be insulin. Transport would be like, something like a membrane protein. Okay. And we know, for example, the sodium potassium pump. Okay. That's the proteins. Lipids, okay. Lipids, this is not really monomers. They're more like components, right? So we have glycerol, right? We have fatty acids. Okay, and then we might have a phosphate group, right? Biological functions, structural support, again. Of cell membrane, we'll say structural integrity. Of cell membrane, signaling, energy storage, so example, for structural integrity, we have the phospholipid. Phospholipids. Also found in the cell membrane are cholesterol. For signaling, it's also hormones. Okay. So example of steroid hormones would be testosterone. Okay, so hormones can either be protein or steroids. We talked about that before. Okay. And then a fat. fats would be energy storage, right? Could be saturated or unsaturated. Okay. That is that. Nucleic acid. So the monomers for nucleic acids would be a nucleotide. We talked about that already, nucleotide. And biological function of nucleic acid. Genetical. Protein synthesis. Example would be like DNA, RNA. So there are different types of RNA, right? Some makes up the ribosome. Some are going to be like an intermediate a messenger, right, between DNA and proteins. We can talk more about that later. Okay. Any question about the macromolecules before we move on? Okay. How is the shape and structure of protein stabilized? I remember we kind of fill in a chart for this, but let's just go through it again. So like if you take the amino acids and you link them together, you get a protein, right? Okay. So the primary structure is basically the order of amino acid. Okay. What holds them together? Amino acids are connected through peptide bond. Right? Peptide bond, it's a type of covalent bond, right? Covalent bond. Right? So this and this link together. Now you might remember that the amino acid looks like this, right? You don't have to know how to draw it, but you should recognize one if I show you one. Okay, so there is going to be the amino group. There is the acid group, right? The carboxylic acid group. And then this is the side chain. Okay, so all 20 amino acids are going to have the same frame structure, same for all 20 amino acids. Okay, same for all amino acid. It's the side chain that's going to differentiate them. It's the side chain that's going to give them unique property. Okay. So that's the primary structure. And then what's going to happen is this thing, after you connect all these amino acids together, each of the circle is an amino acid. After you connect them together, they're going to fold up. Okay, so it's going to fold up into these alpha helices or the beta pleated sheets. Okay, so we have the alpha helix here. And then we have the beta pleated sheets. Okay, so these coils and zigzags, they are part of the secondary structure. Okay, secondary. Okay. And they are stabilized. Stabilized by hydrogen bond. Okay, so I can't really show you clearly on this, but if I zoom in a little bit, right, there will be hydrogen bonds. Choose a nicer color, it's the one that stands out. There will be hydrogen bond all over the place along the coil to stabilize it. Okay, same thing for the zigzag. Okay, all these are hydrogen bonds. Now you might be a little bit confused because earlier I said the hydrogen bonds, it's interaction between two molecules, and then here it seems like it's interaction within the same protein. That could happen if the molecule is really, really big. Okay, so stabilized by hydrogen bonds. Okay, what's gonna happen next? Well, this is gonna then fold up into like a three-dimensional structure, right? Okay, so like, you know, 6Z here. So overall, like a 3D shape object. So the tertiary structure. Okay, overall 3D shape. Okay, stabilized by interactions between the side chains that I talked about earlier. Okay, could be hydrogen bond, could be ionic bond, okay, and a bunch of other possible interactions, okay? But the key thing is it's the interaction between the side chain. Here is stabilized by the hydrogen bonds of the backbone. Backbone, okay? Remember in lecture 2, I think we did this, see? We drew a bunch of amino acids together, right? So this yellow thing is a backbone, right? Okay, so you can see the oxygen here is slightly negative, and then the hydrogen here is slightly positive. So if this backbone, let's see if I can draw this. If this backbone folds back, right up here, right, it just keeps on going like that, and then you happen to have like the same thing, right? Keep drawing the same thing, and then you will have the N here and the X here, right, and the C and the O, right? So this part is now slightly positive, right? Slightly positive, slightly negative, right? Then now they could interact, right? So that's the interaction of the backbone, the hydrogen bond. That's what stabilizes the secondary structure. And for the tertiary structure, it is stabilized by the interaction of the side chain. And finally, some proteins might have the quaternary. Okay, structure. And that's like when multiple peptides come together, right? Okay, so peptide 1, peptide 2, 3, and 4. Okay, several of these things come together and form like a protein complex. Okay, and that would be held together by a bunch of interactions. Again, hydrogen bond, ionic bonds, hydrophobic interactions, so on and so forth. Okay, so that's your protein structure. That is it for lecture 2. We still have a bunch of stuff to do. Let's move on. Cells, that's lecture number 3. List the major components of cell membrane. If that shouldn't be too hard. So first of all, the major component, like the one that is like I would say 90% of the entire membrane would be the phospholipid, right? Phospholipid bilayer. Okay. So we have the hydrophilic head facing the inside as well as the outside. Okay, and then in between them, that's where you have the hydrophobic tails. Okay, so the best way to describe this is hydrophilic heads sandwiching the hydrophobic tails. Okay. Okay, so this is outside. This is inside of the cell. Now, every now and then you're gonna have a protein, right? You're gonna have a protein like this. Sorry, I'm writing all over the place. This is hydrophobic tails, okay? So here is the integral protein. Right? Integral protein. An integral protein spans the entire length of the phospholipid bilayer, right? So an example of an integral protein would be like a channel protein, right? That allows things to like kind of go through. Okay, so that's an integral proteins. Sometimes you would just have a peripheral protein. Peripheral protein. So an example of something like this might be a membrane protein that catalyzes a reaction. So A comes here, and then it will convert it into B. And we learn about this when we discuss the functions of the membrane proteins, right? And let's draw a few more. You might have another protein here that has sugar on it. Right, these are sugar molecules. Right, so that would be like a glycoprotein, which would be useful for cell recognition, right? Okay. And then sometimes you will have like glycolipids. You would have a lipid here, and then you will have like sugar on it. Right? Glycolipids, also useful for like cell signaling or cell recognition. And also you will have some cholesterol here and there, right? Okay, so these orange things, they're the cholesterol. Remember, the cholesterol is important for controlling the fluidity. Controlling fluidity. Okay, too much cholesterol, it'll be very leaky. Too little, it'll be very rigid. You need to have just the right amount. Okay, describe the central dogma of biology. Be sure to mention the related structure, location of in your description. Okay, so basically it's not too difficult. This is the nucleus. Okay, and in the nucleus, you will find your precious DNA. Okay, so that is the DNA. Now, the central dogma, like central theme, right? Central idea, central principle of the biology is that DNA to RNA to protein. Okay, so DNA contains the instruction to make proteins, but we are going to make the proteins in the cytoplasm, right? We don't want DNA to leave the nucleus, so we make a copy of the parts that we need in the form of RNA. So this process is called transcription. Okay, and that happens in the nucleus. So DNA is going to become a copy. RNA. You don't copy the whole thing, okay? You only copy the pages that you want, okay? Think about like, having a textbook at the library, like a study guide or something. You're not going to photocopy everything. That's like impractical, right? Too expensive, takes too much time. You're only going to photocopy the pages that you need. Same thing for DNA. You're not going to copy the entire DNA. You only copy the part that contains the instruction to make the proteins that you need at the moment. Okay, so that RNA is going to leave the nucleus through the nuclear pores, and now it's going to be in the cytoplasm. Okay, there is the RNA. And then what's going to happen is all these ribosomes will come, and then it will decode the RNA to make the polypeptide. Right? So this is the protein. So by using the instruction of RNA to make protein, that is called translation. Okay, so this is the central dogma of biology, and this is where, you know, each step kind of take place. One function of the liver is to remove toxin from blood. Based on this function, which two organelles would you expect to be abundant in the liver cell? Okay. Which two organelles? Anybody? I'm looking for two answers here. Remove toxins from the blood. What two organelles will be abundant in a liver cell? You will find a lot of them. The answer will be lysosome. Okay, so remember, lysosome contains various enzymes, right? Various enzymes to break down molecules, macromolecules. Okay. Toxins, old organelle, right? It might even release all those toxins if the cell gets too old and kill the cell, right? Program cell death. Okay. So if the liver has to remove toxins, you definitely will see a lot of lysosome. The other organelle that you will see a lot is the peroxisome. Okay. Which uses oxidation reaction. You know what that means now. We just learned it on Friday. It strips electrons from things. Uses oxidation reaction to break down toxins and wastes. Okay. Generating H2O2 as a byproduct. Okay. So you have a lot of lysosome as well as peroxisome in liver cells. Describe the structure of mitochondria and state of function of each part. We kind of did that on Friday, but let's talk about that again. Okay, so you have the outer membrane. Outer membrane. Nothing much is going on in the outer membrane. The outer membrane basically forms a barrier between the cytoplasm and the intermembrane space. Intermembrane space. Okay, what's the importance of the intermembrane space? It allows H+ concentration to increase during electron transport chain. Remember, all the hydrogen is gonna get pumped, right? That's why you need the outer membrane, I guess, right? So that the hydrogen is not gonna leak out to the cytoplasm, right? Okay. And then here, this is the cristae. Cristae. Right? It's the foldings of the inner membrane. Okay, so the ETC is embedded within the cristae. Now, why do you want to fold it up? Why not just have, another circle like that, right? Well, by folding it up, right, the foldings drastically increase surface area, right? And by increasing the surface area, you increase reaction space and speed. Right? So you can pack a lot more ETC in the crystay, right? So like there's an ETC, there's an ETC, there's an ETC, right? And then you might have the ATPase here like that. So you can pack a lot more of the ETC if you fold it up. Finally, the inside is the matrix. Okay, so what happens in the matrix? Well, in the matrix, we have the Krebs cycle and we have the pyruvate. Pyruvate oxidation, right, plus Krebs cycle. And also, we can do the oxidative phosphorylation. Oh boy, sorry. There we go. Okay, that's where when the H plus comes back in, you're gonna make ATP, right? So that is the oxidative phosphorylation, part of it anyways. So that's that. You will expect to have a lot of mitochondria in cells that requires lots of energy, like your muscle cells, for example. That's going to have a lot. All right. Describe the endomembrane system. Okay, so endomembrane system. We have our nucleus. The nucleus is surrounded by the nuclear membrane. Okay, so that is the nuclear membrane or nuclear envelope. Okay. Now that nuclear membrane will continue to extend into your cytoplasm. Okay. And it will form the endoplasmic reticulum. So some of the endoplasmic reticulum will have ribosomes on them. Some will not. So these are your rough endoplasmic reticulum. Okay. They work with ribosomes to synthesize and package proteins. Okay. So from the endoplasmic reticulum, you're going to have a bunch of vesicles coming out. Okay. And then the vesicle is going to have like proteins in them. Okay. Vesicle with protein. Now, further out from the nucleus, you're going to have endoplasmic reticulum with no ribosomes. So these are the smoothies, the smooth endoplasmic reticulum. Okay, and they do not do protein synthesis. Okay, their job is to synthesize lipids. Okay, and also breaks down lipids as well. Okay, if there's some kind of fat that you need to get rid of, that's what the smooth ER is responsible for. Okay, still further out from the nucleus, you will have these stacks of membranes. Okay, those are your Golgi apparatus. Okay, they will receive the vesicles. They will package, they will sort, modify, and repackage repackage the vesicles, the proteins, the contents of vesicles. Okay. So this vesicle comes here and then it gets sorted and modified. And then, you have a new vesicle. The content might be a little bit different now. Okay, so where do they go? Okay, where do they go? Well, some will go and be stored in cytoplasm for later use. Some will become lysosome, right? The one that contains the digestive enzymes, can break down things. Some will be released from the cell via exocytosis. You should know what that means by now, right? Okay. And then some will merge with cell membrane. And put a membrane protein there. Okay. So that's the endomembrane system. All right, we're going to take a short break, 5 minutes. We'll come back at 9:37 and we'll do more. Okay, see you in a bit. All right, let's continue. Next, we have Lecture 4, Membrane Transport. Explain what passive transport is, provide examples of passive transport. Okay, so passive transport, there are two key things to know here. Number one, it is moving substances from high concentration to low concentration. So down its concentration gradient, and then therefore no ATP is needed. Okay. So example of passive transport, we have diffusion, okay, osmosis, which is diffusion of water. Okay, remember that a question we did? If you have like a U-shaped tube, and then there is a semi-permeable membrane. We have water on both sides, and then we have solutes like that. Okay. Which way will the water go? Water is going to move this way, right? And it's going to move to the left to try to balance out the concentration. So that is osmosis, and it does not require energy. There is also facilitated diffusion. Okay. And then there are two types. There is the carrier protein, and then there is the channel protein. Okay, so carrier protein is the Pac-Man, right? Remember? The substance binds, and then it causes it to change the shape, causes the protein to change the shape, and then it will transport the material to the inside. Whereas channel protein is just a protein with a tunnel for things to go through. Okay, so facilitated diffusion is crossing the membrane with help of a protein. Okay. Also, no ATP is required. Okay, these are going down its concentration gradient. It's just that they are too big or they are charged, and so there's no way for them to cross the phospholipid bilayer on their own. That's why they need to have the membrane protein. Okay, so those are all the examples of passive transport that we talked about. Next one, describe the differences between various types of bulk transport. Okay, so bulk transport. That just means you have to move a lot of big things in your transport. So there is the exocytosis. Exocytosis. So in exocytosis, you will have your vesicle. I'll draw the vesicle here in red. In red, so you will have your vesicle with your material, and then the vesicle will move towards the membrane. And then what's going to happen is it will fuse with the membrane. It'll fuse with the membrane. Oops. Try that one more time. It will fuse with the membrane like this. Okay. And then afterwards, what's going to happen is it will look like this. Okay, it will become part of the membrane and the material will be released. Okay, so this is exocytosis. And as you can see, you actually increase the surface area of membrane because you're adding a piece to it, right? Okay, this becomes part of the membrane afterwards. So earlier, before the break, we said how some of it can merge with the cell membrane and put a membrane protein there. So I can show you what that looks like, right? Let's say instead of having these little dots here, right? We could have a vesicle, and then we can have a protein stuck to it. Okay, so that's going to move towards the membrane. Right, going to move towards the membrane, and eventually, when it's all fused, and you have an integral protein like this. Okay, so that's exocytosis, moving things out of the cell. The opposite of exocytosis is endocytosis. There are three types. There's phagocytosis, right? Cell eating. So like a cell, and then there's a bacterium, and then you will eat it. That will fuse with the lysosome, which gets broken down. We can also have pinocytosis, which is cell drinking. The main difference is that you will be taking in primarily liquid fluid rather than like a solid. And then the last one is receptor mediated endocytosis. Okay, so the difference between this and the other two is, in order for you to bring the material in, they have to first bind to a receptor. So this is more specific. Okay, so for example, here's a cell, and on the surface there is a little receptor here. Okay, so if I have these little circle things that bump into the receptor, they will not be taken in. Only the triangles will be taken in. So when triangle binds, it will cause the area to kind of cave in, right? And then afterwards, it will be something that looks like this, right? Along with the receptor, the material will be taken in like that. Okay, so it requires a receptor, and it's going to be more specific because whatever you're taking in, whatever triggered this event, must fit in the receptor. Okay, so those are the types of bulk transport. Of course, bulk transport requires ATP. It's a type of active transport, a type of active transport. All right, that is that. Number 7. A cell of a plant that grows in very cold environment was studied. What kind of adaptation do you expect the cell membrane to have? Okay, so a couple of things here. At cold temperature, the cell membrane becomes more, does it become more fluid or does it become more rigid? What do you think? Based on our, lab, do you think at cold temperature, the cell membrane becomes more fluid or rigid?
More fluid.
More fluid means more leaky, right? So is that what's going to happen when it's cold?
Oh, it will be rigid then.
Yeah, exactly. It's more rigid, right? It becomes more rigid, right? More rigid, which is lower permeability. Okay, so that's the first thing to realize. Okay, as you increase the temperature, cell membrane becomes more fluid, and then they are going to become more leaky. So if we have a plant that grows in very cold environment all the time, right? We don't want the cell membrane to be too rigid because then nothing goes out, nothing comes in, right? So what kind of adaptation do you think is going to happen? Well, I think, yeah, go ahead. Did you have a suggestion, Yusuf?
Can I have a question? Can I ask a question, please?
Yeah, of course.
Instead of cold temperature, if it is a freezing temperature, would that have an effect? Is it going to be lower permeability or higher permeability?
Say that one more time, please. I didn't catch the question.
Okay, instead of cold temperature, if it is a freezing temperature, I think in the bit.
Okay, I understand your question now. Okay, so you're saying if it's freezing temperature, is it going to increase the permeability? Now,
yeah.
It's a tricky thing to talk about. If it's a plant that goes in a freezing temperature, based on what we talk about in class, right? You are thinking if it freezes, the crystal will kill the cell, will puncture the cell membrane, and then it will become very leaky, right? Is that your logic?
Yes.
Yes. However, we must understand in nature, even though if it's freezing, the ice crystal should not be able to puncture the cell membrane, because if that's the case, then all the plants will die in the winter, right? But of course they don't, because the next spring they would still be alive, right? So there must be some kind of adaptation, some kind of special features that allows them to not experience this. So in the case of the ice crystal, there would be like natural antifreeze in the plant that would prevent the ice crystal from forming. Okay.
Okay.
And that's what this question is asking, right? At cold temperature, what kind of adaptation would you expect? So in order to prevent the cell membrane from becoming more rigid, you would, for example, expect to have higher percentage of unsaturated fatty acid tails in the phospholipid, right? Remember, if it's unsaturated fatty acids, then they don't pack as easily, right? Because of the kink, remember, right? There's the kink. So if all of them has more unsaturated fatty acid tail, then the phospholipids will not pack as easily, even at cold temperature. So that's probably one of the adaptation, right? This prevents the phospholipids from packing together closely, even at lower temperature, right? For example, you put a piece of butter into the fridge, like after you melt the butter and then you put it in the fridge, it will become solid, right? Okay. But if you put like oil, like cooking oil into the fridge, it's not gonna become solid, right? It might become a thicker, but it's not gonna become like solid, right? And that's because the cooking oil, the plant oil, it's going to have a lot of unsaturated fat. It's basically unsaturated fat, and they don't pack easily. Okay. Another adaptation is you would expect to have higher percent of cholesterol in the membrane. Okay, remember cholesterol increases fluidity. Okay. Now, conversely, if you compare this with like a plant that grows in the desert or something like that, it will be completely opposite. You would expect to have fewer cholesterol. You would expect to have more saturated fatty acid. Okay, so I hope that makes sense. Please remember that. Next one, how are active transport similar to facilitated diffusion, and how are they different? Okay, they are similar because both require membrane protein. Okay, so active transport, when we're talking about like the sodium potassium pump, that requires a membrane protein. Facilitated diffusion, right? The Pac-Man that I talk about, and then the tunnel, those are membrane proteins. How are they different? How are they different? Well, active transport requires ATP, and it's going from low to high concentration. Whereas facilitated diffusion does not require ATP because it's passive transport, and it goes from high concentration to low concentration. Right? So that is that for lecture number 4. Okay. Lecture 5, which is what we did on Friday. What is an organic molecule? Organic molecule is any molecule containing carbon. Okay. Except there are a few exceptions, except for CO2, right? And CN and CO. These ones, we don't call them organic. Everything else that has carbon is organic. Okay, so all the macromolecules that we talked about, they have carbon in it. They are organic. Okay, that's basically it. List the products of glycolysis. Okay, so glycolysis, right? We will say what goes in and what comes out. Okay, so what goes in? Well, what goes in is we need to have ATP, right? The 2 ATPs to prime the reaction to energize, right? To supply energy, the energy investing phase. We're going to need to put 2 NAD in as well, right? These are things that goes into glycolysis. And we also need to have 4 ADP. Okay. So what's going to happen is afterwards, these ATPs will become 2 ADP. Okay. The NAD will become NADH. The 4 ADP will then pick up the phosphate release from this glycolysis process and becomes 2 ATP. Okay. So these are the major products. And of course, the 2 pyruvate. You got to give them a glucose as well. Glucose. And then they will become 2 pyruvate. Okay, so these are the major products. Okay, so you invested 2 ATPs. Sorry, this should be a 4. And then you produce 4 ATP. So overall, it's 2 net ATP. And may I remind you, after glycolysis, most of the energy is still locked in the pyruvate. And in the presence of oxygen, this will go on and be further broken down. Otherwise, it will undergo fermentation. Okay. Next. Hold on. In cellular respiration, glucose is oxidized while oxygen is reduced. What does it mean? So glucose is oxidized. It means electrons are removed from glucose. Okay, and remember the electrons are captured by NAD or FAD. NAD plus and FAD. And oxygen is reduced. It means electrons are added to oxygen to become H2O. Okay, so it's a redox reaction. It's a transfer of electron from glucose essentially to oxygen. What is the role of NADH and FADH2 in cellular respirations, right? They capture all the electrons released from glycolysis, pyruvate oxidation, and Krebs cycle. The electrons are transported to the ETC, where, where the energy is used to pump H plus from the matrix into the intermembrane space. What is the significance of adding coenzyme A to pyruvate during pyruvate oxidation? Remember, pyruvate oxidation, three things happen, right? You remove a carbon dioxide, you strip some more electrons from it, and then you're going to add a coenzyme A. The purpose of the coenzyme A is to coenzyme A creates an energetic and unstable bond such that the molecule is ready to combine with oxaloacetate. In the Krebs cycle, okay, such that acetyl-CoA, right? Acetyl-CoA is ready to combine with oxaloacetate in the Krebs cycle. Okay. All right, here is a math question, the math question that we sort of did on Friday. Assuming a mitochondria contains 50 NADH and 19 FADH2, if energy carriers were used, what is the maximum number of ATP produced? So 50 NADH times 3, right? That's 150 ATPs. Whereas 19 FADH2 times 2, That's 38 ATPs, right? Each NADH can produce 3, and each FADH2 can produce 2. And then you add these up together, that's going to be 188 ATPs maximum. The inner mitochondrial membrane area is about 5 times the area of the outer mitochondrial membrane. What is the purpose of this? Right? So we talked about this already, right? It increases surface area for ETC reactions, right? You can pack more ETCs in it. And the more ETCs you have, the faster you'll be able to pump H+ across the membrane to generate a gradient, which increases your ATP production step. Which steps of cellular respiration generate the greatest number of ATP? It is the electron transport chain, right? Okay, at the ETC, majority of ATP is produced here through oxidative phosphorylation, right? Right? And also chemical osmosis, right? Creating that hydrogen gradient. Describe what happens as H+ is pumped from matrix into the intermembrane space. Okay, so a couple of things going to happen. Right? This is the cristae. Right? As we pump H+ into the intermembrane space, you're going to create a hydrogen gradient. So you will have high H+ here compared to low H+. They really want to come back in. Right? They really want to come back in. Now, because of the high H+ content here, you're going to have a lower pH in the intermembrane space. You're gonna have a lower pH, more acidic in the intermembrane space, and you're gonna have higher pH in the matrix. Okay. And the purpose of the final electron acceptor, okay, the final electron acceptor is oxygen, O2. Again, the purpose of that is to remove electrons from the last protein on the ETC. Okay, it's like I think I talked about this on Friday. It's like slide at the park, right? If the kid at the bottom does not go away, then the top one cannot slide down. So the parent's gonna come, it's gonna take the kid away so that the next one can go. Okay, so in a similar way, you have your ETC proteins, and as the electron get passed down, the oxygen will pull the electron away from the last protein on the ETC, and that allows the next one to slide down. Okay, so that's it for the review. Let's go back to the what was this? The study guide for lecture 5. And let's talk about the application questions and fill in the blanks, and that will be it. Okay, so I'll give you a moment to pull that out. Okay, so cyanide has a higher electronegativity than oxygen, which means it attracts electron better than oxygen. In cyanide poisoning, cyanide binds to one of the membrane proteins in ETC. Explain how this could result in death very quickly. Okay, so as I just mentioned earlier, right, the electron's going to get passed down. Okay, And then here is the oxygen that will pull the electron away, right? So the reason why oxygen can pull the electron away is that the oxygen has a higher electronegativity, okay? Higher electronegativity compared to the ETC protein. Okay, think of them as like magnets, okay? Each of these are magnets, and then you're passing down like a paper clip or something. In order for the second protein, the second magnet to pull the paper clip from the first, the second one has to be a stronger magnet, right? And the third one has to be a stronger magnet. And finally, if you want to pull the paper clip from this magnet, you will need an even stronger magnet, right? So oxygen is like the strongest magnet in all of these things, okay? And you need to remove the electron from the ETC so that the next electron can slide down, right? If the ETC is clogged up, then you won't be able to pass down the electrons, and you won't be able to pump H+. Without the H+ gradient, you will not be able to produce ATP. That's how you die. So what happens is if you have cyanide, and cyanide is going to be binding to one of the membrane proteins in the ETC. So let's say cyanide binds here. And this has a higher electronegativity than oxygen, which means O2 is no longer the strongest magnet, so to speak. And the electron will be stuck to the cyanide, and O2 will not be able to remove them. As a result, the entire ETC is clogged, right? You can no longer deposit electrons on it, right? So you have an NADH coming here, but it cannot put more electrons on it because it's full, right? And therefore, you won't be able to generate the H+ gradient. You won't be able to produce ATP, and you die. Speculate what would happen if toxin disrupt the integrity of the inner mitochondrial membrane. So here is the outer membrane, and here is the inner mitochondrial membrane, right? We pump H+ through it, and then the H+ cannot come back in because it's charged, right? It will have to go through the ATPase. But in this case, they're saying what happens if we disrupt the integrity of the inner mitochondrial membrane? So, you know, there is a bunch of holes in it. Well, then the H+ is just going to leak right back through without going through the ATPase. And if that happens, then you are not going to be able to generate ATP at maximum capacity, right? So, you know, that's going to be a problem. You will not have enough ATP for your cells. Okay, let's quickly fill this in. The first step in cellular respiration is glycolysis. This process takes place in the cytoplasm of a cell. A glucose molecule is split into two molecules of pyruvate. To initiate the process, two ATP molecules need to be invested first. The process generates 4 ATPs in total. So the net ATP production is just 2. All the ATPs generated in glycolysis is through a process called substrate level phosphorylation. This requires an enzyme that transfers a phosphate from a fuel molecule or donor molecule onto ADP. As glucose is oxidized, high energy electrons are released. They are captured by energy carrier called NAD+. There's no FAD yet. FAD only shows up in the Krebs cycle. NAD+ captures the electrons and combines with 2 hydrogen ions to form NADH. The NADH will take these high energy electrons to the electron transport chain. Where it is used to generate ATP. Okay. pyruvate oxidation. If oxygen is available, the two molecules of pyruvate generated in glycolysis will proceed to the next stage in cellular respiration. This stage is called pyruvate oxidation. And it takes place in the matrix of the mitochondria. There are 3 steps here. Carbon dioxide is removed. High energy electrons are captured by more NAD+, creating NADH. A helper molecule called coenzyme A is added to pyruvate, converting it into acetyl-CoA. Okay. Next is the citric acid cycle. Okay, so the next step in cellular respiration is called citric acid cycle, which is also known as the Krebs cycle. The product in pyruvate oxidation is acetyl-CoA. This molecule, acetyl-CoA, combines with oxaloacetate in the matrix of the mitochondria. So the acetyl-CoA, which is 2 carbons, will combine with a 4-carbon oxaloacetate to form citric acid. And then citric acid is broken down into carbon dioxide, which is released as product of cellular respiration. In the process, high energy electrons are captured by NAD+ again, as well as FAD, generating NADH and FADH2. The difference between these 2 energy carriers is that FADH2 has lower energy than NADH. Right? This can only produce 2 ATPs, while this can produce 3 ATP. In addition, ATP is also produced in the Krebs cycle by substrate level phosphorylation. At the end of the cycle, oxaloacetate, the 4-carbon compound, is regenerated so that it can combine with another molecule of acetyl-CoA. Last but not least, we have the electron transport chain. The last step in cellular respiration is called the ETC. It takes place in the matrix of the mitochondria. Oh, sorry, not the matrix, the cristae. The cristae of the mitochondria, which are formed from the folding of the inner membrane. Embedded in the inner membrane are a series of membrane proteins. These proteins are responsible for transferring electrons that are deposited by NADH and FADH2. As the electron flows down the protein series, it loses energy. The energy is used to pump H+ from the matrix into the intermembrane space. At the end of the protein series, oxygen is required to capture the electron to form water. This frees up the ETC. As H+ is pumped from intermembrane space, it generates a high concentration of H+. However, since the inner membrane is impermeable to H+, they cannot diffuse back into the matrix. A special proteins called ATPase allows H+ to diffuse from intermembrane space into the matrix. As H+ passes through ATP synthase, the energy is used to add phosphate onto ADP generating a large amount of ATP. So that's that. That's the fill in the blanks. I went through it relatively quick because I assume you have done it already. And yeah, that's it for the test review. So the test is on Friday. Make sure you don't miss it. It's going to be available from 6 to 11:59. And if you have any question between now and then, please feel free to email me. And of course, there is also the practice self quiz on eCentennial that you can do to study. Yeah, so that's it. Okay, thanks for coming. I'll see you all.
We are not meeting on Friday, or are we meeting?
No, Friday is just the test. So our next tutorial will be next Friday. Okay. Okay, have a good one, guys. See you all later.